Hi, ladies and gentlemen. We are going to begin this learning adventure today, talking about monohybrid crosses. Our two aims for today are to describe Punnett squares and their importance, and also to be able to solve and describe the outcomes of monohybrid crosses. Just a screen to remind us of our true-false question for this particular CC. And this true-false question is when a specific gene separates in meiosis, the alleles for that gamete separate from one another. Now, we're coming back to our homologous chromosomes. So once again, let's just say that this is chromosome number 1. You got this homologous chromosome from your mom, and you received this one from your dad, or this particular individual, I should say. And on this chromosome, we're going to have the same gene. So let's say this is not indeed your chromosomes, but the chromosomes from a pea plant. So this is the gene for flower color. So G1 equals, gene 1 equals flower color. And remember that on this homolog from our mom, we could have a big P, meaning the allele is purple flower color, or we could have the little p, which is white flower color. So this particular pea plant cannot give both of these alleles to its offspring, and that is the law of segregation. So it is going to either give this allele or it's going to give this allele to its offspring through the process of meiosis, which is making of an egg or a sperm cell. A reminder, this is why we started with meiosis, that parents, through sexual reproduction, parents can only give their child half of their genetic material or half of their chromosomes. Humans, in our somatic cells, we have 46 chromosomes. However, in our sex cells, in our gametes, we only have half that genetic information, or 23 chromosomes. Meiosis is the process by which we go from a diploid cell to that haploid cell. Let's take a look at a couple terms here and make sure that we have them square in our notes. So if you need to add some additional things or highlight, you should be doing that right now with your 11.2. So the first term, once again, is genes. And remember, a gene is a sequence of DNA that codes for a trait or codes for a particular protein. And because it codes for that protein, it determines a trait, whether that's a specific color, whether that's a specific height, could be a number of things. One example is eye color, so either blue or brown eyes. Now, with that gene, we have two alleles for most genes, and those alleles are different versions of the gene. So first of all, our gene, once again, is for eye color, and then our different versions could be blue or brown. Once again, we designate the alleles with letters. And actually, brown is a big B and blue is a little b. Now, I'm going to have a link to this video, what is a gene, right in the Canvas site, so you can take a look at that for that clarification as well. Once again, taking a look at a pair of homologous chromosomes. Once again, this one came from this individual's mom. This one came from this individual's dad. The gene that's being coded for on these particular chromosomes is flower color. So this particular homolog has the allele for purple flower colors, and this one has the allele for white flower colors. You get one allele from your mom, you get one allele from your dad. And once again, when you go through the process of meiosis, you can only give one of these alleles to your offspring. So once again, here's our terminology. A dominant trait is a trait that can hide the appearance of another trait, and we represent that by capital letters. So the big capital B represents the allele for brown eyes. Once again, that is a allele. And the recessive trait is a trait that can be hidden by another trait, and it's represented by lowercase letters. Now, this is the infamous song that you heard in middle school. Girl, your phenotype is showing. All right, so phenotype is the physical characteristics. What does it look like? Now with phenotype, you can see down here on the lower part of my slide here, I've got two different phenotypes. I can either have brown eyes or I can have blue eyes. And we have to remember our rules of dominance once again. Anytime you have a dominant allele, that allele is going to be expressed. So I have the genotype big B, but it's brown eyes. But I also have a different genotype, which is, once again, the genetic makeup. What are the alleles for that particular individual? And when I have a big B and a little b, so that's a different genotype than what I had up here, but the big B masks the recessive allele, therefore I still get brown eyes. And once again, in order to get the recessive phenotype, I need two little or recessive alleles. So when I have little b, then I get the blue phenotype. So two other important terms we need to know are heterozygous and homozygous. Now hetero, that root word, means different. And the root homo means same. So if we have different alleles, then we have, and it can be any letter combination, but it has to be the same letters. We can have big H, big T, little r, big R, little r. This is also referred to as hybrid, so that's important to note. If you're a hybrid, you're a heterozygous individual. You have two different alleles. All right, so X, little x, that's a heterozygous individual. Now, looking down at homozygous, we have two of the same alleles. And you can either be homozygous dominant, so big A is homozygous dominant, or if I have two little a's, that's homozygous recessive. As long as you have the same alleles, it's homozygous. This is also referred to as true breeding or purebred. So if you ever see a problem that asks for those two, you know that it's homozygous. And just a little bit of humor. You can pause and enjoy and chuckle a little. So let's take a look at this problem from your homework. You were to label the parental generation. And this one is our parental generation. And you'll notice that we have crossed a homozygous dominant by a heterozygous dominant. And when we do that, we get 100% of the offspring here are heterozygous. So they have the dominant allele and they have one of the recessive allele. So these are heterozygous. And looking at your CC, this is also called the filial 1 generation. And this is going to be our filial 2 generation. So when we take our homozygous dominant by our homozygous recessive, once again, homozygous, same alleles. Okay, we have two of the small alleles over here. And we cross that. The only allele that this parent can give to its offspring is the dominant allele. The only one this can give is the recessive. So that's why we get 100% heterozygous for that filial 1 generation. Now, if we do this, the only option is to take a heterozygous by a heterozygous. So if this particular plant were to self-pollinate, and this is where we start to get our Punnett squares. So this particular heterozygous individual has two options in terms of gametes that it can give its offspring. It can give the dominant allele or it can give the recessive allele in each of its gametes. So when we do a Punnett square, we are showing fertilization possibilities. So that's what we're going to put across the top of our Punnett square, that recessive allele versus the dominant allele. Once again, it's the same thing for this individual. It could give the dominant allele to its offspring or to its gamete, or it could give this one. So the Punnett square shows us all the different possibilities of offspring. And in this particular one, we've got a 25% chance of a homozygous dominant. We get a 50% chance of a heterozygous, and we get a 25% chance of a homozygous recessive. If we were to look at phenotype, this is genotype. Remember, when we're looking at alleles, that's genotype. If we were to look at the phenotype, okay, this one is going to be that dominant, which is tall. This is also going to be expressed as tall, and this is also going to be expressed as tall. So it's 75% tall, and this is our only plant right down here. And so that's 1 out of 4, or 25% of the offspring are short. So a monohybrid cross is simply a cross between organisms that differ only in one characteristic. And this is one of the things that Mendel did that was different than his predecessors, is he looked at one characteristic at a time, whether that be tall versus short, purple versus white. All right, he looked at just one characteristic, mono, meaning one. All right. And this is another video that I will post to the Canvas site, and I think it's worth your time watching, just a couple of minutes, and you can take a look at Mendel's peas. So it's important to remember what Punnett squares do. And what Punnett squares do is they help us find the probabilities. All right, they don't tell us what is exactly going to happen. It's the chance that a specific event will happen. It's like if we flip a coin. All right, if I flip a coin 10 times, okay, you could tell me that you're going to get heads 5 times and you're going to get tails 5 times. All right, so let's actually do this. And you could do this alongside me as well. And so we know based on probability that we should get 5 tails and 5 heads. However, if we do this, so I'm going to flip right here, I get 1 heads. All right, flip again. I got another heads. I got a tails. Got another heads. Flip again. Heads. Heads. Tails. Tails. What am I at? 8, 9, and 10. So looks like I ended up with 7 heads and 3 tails. If I were to continue to do this, maybe I would get, and over time and getting a lot of data and a lot of flips, I'd probably equal about 50% heads, 50% tails. But I want you to remember this about a Punnett square, is that when we look at the likelihood of each potential offspring, it's simply a probability. Right? It doesn't say that we have to get, if we have 4 offspring, that only one of the 4 is going to have a homozygous recessive phenotype or recessive genotype as well. It's simply what is likely or the chance that something will occur. All right? Yes, if you have 4 children, you would maybe expect, based on probability, that you may have 2 girls, 2 boys, but you could always end up with 4 boys, 0 girls. All right, it's the likelihood. All right, the last thing that's really important is to figure out how we can report out our results. So you are going to want to make sure that you have this written somewhere in your CC. There are 3 different ways that you can report your results, and I don't really care which way you do it, as long as you know how to interpret each of these when you see them. The first is ratios, okay? When we're talking about reporting out phenotypes with ratios, we always use the dominant first and then the recessive. So with ratios, you should make sure that all of your numbers should equal the total number of squares. So with what we see when we cross 2 heterozygotes is that we usually get a 3-to-1 ratio. That means that we have 3 of the dominant trait and 1 of the recessive trait. All right. When we are reporting out genotypes, we always report out the homozygous dominant first. So that'd be one homozygous dominant, the heterozygous second, and then the homozygous third. So if I just quickly do this, all right, this would explain this particular ratio. So I have one homozygous dominant, and that's my one there. I have two heterozygotes, and then I have one homozygous recessive. Once again, if I look at this and I say that big A is tall, little a equals short, if I look here, these three have my dominant phenotype, and this particular one has my recessive. There I get my 3-to-1 ratio. In the same way, you can report out percent. So in this particular situation, if I look at the dominant phenotype, that's 75%, and the recessive phenotype would be 25%. And then finally, the fraction, 3/4 and 1/4. Which way you report out results, I don't care in your homework. As long as if you ever saw the phenotypic or genotypic ratios, you would know how to interpret them or report your results as well. All right, so let's look at an example. This is in your homework. All right, so we're going to say big T is tall, little t is short. We're going to cross two heterozygote individuals. So the way that we set up our Punnett square is this is parent 1, this is parent 2. We're going to put parent 1 across the top. So once again, the reason we're setting up this Punnett square is that with a heterozygote individual, it has the possibility of giving this allele to its offspring through the law of segregation, or it has the possibility of giving that particular allele to its offspring. So if parent 1 equals the male, we can say that these two are possible sperm outcome. And this side is going to be parent 2, and this is going to be our female, and these are possible eggs. So then, when we look down into our Punnett square, this is a chance fertilization event. If this egg gets fertilized by this sperm, we get a homozygous dominant individual. If this particular egg gets fertilized by this sperm, we get a heterozygote along here. And then finally, if this egg over here gets fertilized with this sperm, we get a homozygous recessive individual. So our offspring genotypes, we could say 75% tall, all right? Or if we gave a ratio, once again, it would be the 3-to-1 to 3-to-1 ratio, one of the homozygous dominant. Oops, I made a mistake there. We want to equal a total of 4, 1 to 2-to-1 ratio. Our phenotype, we have 25% short, and that would be a 3-to-1 ratio. All right, the last thing to go through is just some simple steps to help solve your genetics problems with monohybrids as you go on to dihybrids, and then the exceptions to Mendelian genetics. I would make sure that I get this into that little square on your monohybrid problems that you should have imported into Notability. The first thing is you want to make sure you code the problem. What kind of problem are you solving? Is it a monohybrid? Is it a dihybrid? Or with the exceptions to Mendelian genetics, you have to figure out what are the rules for that particular problem. I would say every single time, make sure that you write down the genotypes of the parents. When people try to do this in their head is when they make mistakes. And especially with dihybrid problems, you don't want to make those big mistakes, as that's a lot of time that you need to go back and do. All right, number 3 is determine the possible gametes. When I say determine possible gametes, once again, that is figuring out which are the possible alleles that they can give to their offspring. You're going to make your Punnett square okay, with your gametes across the top. It's again like this. And then you report your results, either in percent, within fraction, or the ratio. So now you may begin your monohybrid cross problems. Once you are done, you are going to upload them to the Canvas page, the monohybrid assignment page, and then you will be given the answers. Please make sure that you check through your own answers. If you have any questions, ask before you move on to the quiz.